Welcome. We're going to continue looking at milk composition. In particular, we're going to start looking a little bit at more specifically at the synthesis of some of the components of milk. In particular, in this video, we're talking about lactose synthesis and secretion. To get started, let me go back and remind you of what the cell looks like. So this is an electron micrograph of a mammary epithelial cell. We've got the nucleus down here. The basal side would be down here where all the capillaries and those kinds of things are. Lumen up here, so this would be milk up in this area. Some large secretory vesicles, we'll get into those here in just a few moments. There's a fat droplet being secreted. So again, this is just simply to kind of remind you of what the cell looks like. Now what I've done is to kind of make a schematic or diagram of this to help us understand how is lactose produced in the cell. So let's go to the next slide. So again, we have the lumen up here. Here's the cell. And this square is essentially a cell. There's another cell here. Another cell here, which I'm kind of standing in the middle of. The basal side is down here, so the capillaries, blood flowing through the capillaries would be in this area, and then again, components would come, precursors would come into the cell, something would happen to them, and they would end up in the milk in the lumen. So I've identified several key organelles in the tissue. Let's start with the tight junctions here. Remember, those epithelial cells have a tight junction between them, typically towards the apical side of the cell. So when we talk about precursors, for example, being taken up by the basal side, we're really talking about what they call the basolateral side. That is the basal side of the cell, basal membrane of the cell, the lateral side, to distinguish that from the apical membrane up here. So again, the cell is polarized. Apical membrane has a little bit different functionality than the basolateral membrane. We also have the nucleus. We have Golgi apparatus, and that's particularly going to become important here for lactose synthesis as well as protein synthesis. These big circles represent the secretory vesicles, and we'll get into those here more in a moment. And then everything else, for our purposes at the moment at any rate, we're going to call cytosol. There are many, many other organelles in the tissue, mitochondria and so on, and we're not going to really talk about those at this particular point. In the mammary epithelial cell, and as in all of our cells in our body, we have an enzyme called galactosyltransferase. And that activity of galactosyltransferase is usually found in the Golgi apparatus. It's involved in glycosylation of proteins, in the process of glycosylation of proteins. And in and of itself, galactosyltransferase doesn't directly relate to milk synthesis other than glycosylating some of the milk proteins, those that are glycosylated. It doesn't really directly involve itself with lactose synthesis. However, when we have a milk-specific protein produced in the mammary epithelial cell called alpha-lactalbumin, those complex, alpha-lactalbumin is synthesized in response to lactogenic hormones, and so it's controlled and regulated in that way. So we see it being expressed during lactation. Those two combine together, and this enzyme galactosyltransferase, the reaction that occurs is slightly modified so that now instead of causing glycosylation of proteins, you take a galactosyl transferase, and combine it to a glucose, which is lactose. We'll get to that here in just a second. So this complex, little indicated here by a little purple, you can just barely see the purple there, complex with the green bar is representing galactosyltransferase, is called lactose synthetase. Lactose synthetase, and those two again combine together to produce lactose. So let's take a little bit closer look at that. So what we have monosaccharides. Again, lactose is a disaccharide, galactose and glucose. I've indicated those here. Glucose is taken across the cell membrane, the basolateral membrane, by transport mechanisms that take it across that membrane. Once it's inside the cell in the cytosol, what's going to happen is some of glucose stays as glucose. Some of the other glucose, though, is epimerized. The reaction is called, the enzyme is called an epimerase. Epimerized to galactose. So they're very, very similar in many respects, just flipping the hydroxyl group in one case. So now we end up with galactose. Glucose is the yellow. Galactose is the white here, I've just indicated. And those are in the cytosol. There's certainly more to it than this, so I'm being simplifying this. Those are taken up into the Golgi apparatus. So we have our precursors, the galactose and glucose inside the Golgi apparatus lumen. We have our lactose synthetase enzyme activity. Let's go to the next slide. That results in putting together, covalently linking the galactose and the glucose. And so that's indicated by the kind of double yellow and white double oval here. That's our lactose. That occurs in the Golgi apparatus lumen. It also continues to occur in the secretory vesicles as they butt off of the Golgi apparatus, as well as they're transported up to the apical membrane. When these secretory vesicles reach the apical membrane, the membranes fuse, a hole opens up, and the contents of the secretory vesicle then is released out into the lumen. That becomes the milk. Let's go to the next slide. The key to this is that as this disaccharide is being produced, water is being drawn in. So these monosaccharides, the galactose and glucose, are not, they can go back and forth across those membranes, the Golgi apparatus membrane, for example. One way or the other, they can be transported back and forth, same way in the secretory vesicle membrane. They're not involved very directly in terms of the osmolality of the solutions inside or outside of this membrane. But the disaccharide lactose cannot diffuse across this membrane, cannot diffuse across that membrane, cannot diffuse across this membrane. As a consequence, water's drawn in to try to dilute that, basically. Let's go to the next slide. So what we have is lactose is considered the major osmole in milk. Now the word osmole in this case is sometimes used is called a unit of osmotic pressure. What we're talking about is something a little bit more physiological than this unit of osmotic pressure. Let's go to the next slide. And so it's really, osmole in this particular case is a volume regulating organic solute. And in the case of milk, lactose, this sugar, this disaccharide, is the major osmole that we find in milk. There are other osmoles, other things that contribute to the osmotic balance across these different membranes, but far and away lactose is the major osmole. So let's do a real quick review here of what we just kind of gone over. Again, glucose from the bloodstream is taken across this membrane, transported across the membrane. Some of the glucose is epimerized, changed into galactose. Galactose and glucose can then be transported into the Golgi apparatus lumen. They are then covalently linked in the Golgi apparatus by the lactose synthetase, the galactosyltransferase and alpha-lactalbumin complex. The production of that disaccharide causes water to be drawn in, and so the water, the lactose, again, by this mechanism of secretory vesicles, is then eventually released into the lumen, and that's how we get lactose being produced.